Good evening, everyone, and welcome to the 2024 Allan and Maria Myers Lecture. I'm Professor Peter van Wijngaarden. I'm Chief Executive Officer of the Florey, and I'm just delighted to welcome you all here this evening. And for those of you who are joining the live stream online, welcome, and I hope you enjoy the proceedings. I'd like to begin by acknowledging the traditional owners of the lands on which we're meeting this evening, the Wurundjeri people of the Kulin Nation. I pay my respects to their elders past, present and emerging. I also extend that respect to Aboriginal and Torres Strait Islander people that are joining us this evening. So we're tremendously grateful to Allan and Maria Myers, who've generously supported this annual keynote lecture. Allan and Maria are committed to bringing the very brightest scientific minds to the Florey to promote collaboration and scientific exchange. And so I'm thrilled that the 2024 Allan and Maria Myers International Visiting Fellow is none other than Professor Nadia Rosenthal. Professor Rosenthal obtained her PhD from Harvard Medical School and trained as a postdoctoral fellow at the National Institutes of Health before leading a highly productive research laboratory at Harvard Medical School. Professor Rosenthal next served as founder and director of the European Molecular Biology Laboratory campus in Rome, before founding and leading the Australian Regenerative Medicine Institute at Monash University, which was one of only 5 regenerative medicine institutes in the world at the time. And I understand that the vision for that institute was entirely Nadia's doing, and she generated a tremendous amount of investment in the infrastructure. She built the place and built it into the stellar institute that it is today. So I think you'll see a thread in that narrative of a founder and director, which really is extraordinary, and I think you'll see her tremendous capabilities during the lecture tonight. So she currently serves as scientific director of the Jackson Laboratory in Bar Harbor, Maine, where she's the Maxine Grosky Endowed Chair. Nadia is Fellow of esteemed academies, including the Academy of Medical Sciences of the United Kingdom and the Australian Academy of Health and Medical Sciences. She also holds a chair in cardiovascular sciences at Imperial College London. Professor Rosenthal is globally recognised as a leader in mammalian genetics, and her research has advanced regenerative medicine by elucidating the role of growth factors, stem cells and the immune system in healing after injury. Nadia has been a pioneer in the use of advanced genetic methods to investigate mammalian development, disease, repair, and particularly in cardiovascular and skeletal muscle. So I've had the pleasure of getting to know Nadia over the last 3 days. She's been a tremendously valued member of our external scientific review panel, and we have other members of the panel here, and I'd like to take this opportunity to thank you all for your time, intelligence, and generosity in coming to provide an external appraisal of what we're doing here at the Florey and really guiding our strategy into the next decade. So over the course of those last 3 days, I've really enjoyed meeting you all, and I know that we have a tremendous amount to look forward to from Nadia in her presentation. So please join me in welcoming Professor Nadia Rosenthal to the lectern.
Thank you, Peter. That was an over-the-top introduction, and I'm not sure I can live up to it. I'm also aware of the fact that probably everybody in this room has been through a very grueling 3 days. For those of us on the interrogation side of the table, it was really no less stressful than for those of you who were being interrogated. And so I'm hoping that the content of this will amuse you, relax you, and essentially get your mind off work for a little while. So, without any more ado, I'm just going to get started. There's something where to go. I thought what I would do is to actually give you a slight a sense of where I am now, given that the last time I was in this auditorium, it was to hear a fabulous lecture by, I can't remember whom, but there have been several. And it occurred to me that you might wonder what the hell I did leaving Australia to go to Bar Harbor, Maine. So I think I just give you a little bit of history about the Jackson Laboratory, because for those of you who work in the mouse, of course, you know it well because it's where you get your C57 black 6 mice and your 129 mice and your, whatever your particular exciting mouse model is of the moment. But it all started, I would like to say, by a woman named Abby Lathrop, who lived in a lovely house in Granville, Massachusetts, where she worked on breeding various animals. She started out with chickens and decided they were too smelly. She went on to guinea pigs, but they took too long to breed, and she then proceeded to mice. But she was a real geneticist at heart because she kept meticulous records of the pedigrees. And so as a result, when she found a mouse that was actually peculiar, she would be able to follow its provenance in a very systematic way. And so I like to think of the Jackson Laboratory as having been started by this very unassuming lady, and that is not a mouse, that is indeed a guinea pig. So at the barn housed 11,000 mice, which is impressive. I know how much that takes to do from being at the Jacks, so I guess it was quite crowded. Now, it all started, the whole way in which the Jackson Lab started was by the fact that she was a good colleague with William Castle, who was at Harvard at the time, and he had an interest in a number of different diseases, including cancer, and started, so she would call him whenever she had a funny mouse. And in this case, she found a mouse that seemed, a strain that seemed to be much more susceptible to tumors. And so she called up William and said, "You might want to have a look." And he said, "Fine." And she sold the mouse by the pound. So you sort of put, I guess, a pound of mice in a bag and sent them over to Harvard. He happened to have two brilliant people in his laboratory. One was C.C. Little, who was interested in cancer as a genetic disease. And remember, this is 1920s. Did we know anything about DNA? We did not. Did we really understand anything about cancer? We did not. So I consider this to be prescient. I mean, I don't know that I would have considered cancer as a genetic disease, but of course the mice didn't hurt. The other person who he had was a much quieter fellow named George Snell, who was working on linkage analysis and used Abby's mice as well. So this went on. C.C. Little, as he was known, went on to be the president of the University of Maine at age 33, the youngest president to ever run a university in America at the time. He built a lab for mouse genetics in Bar Harbor in 1929, and he really was an enormously sort of influential character who was extremely popular with the ladies. He looked like Errol Flynn. And so he used to get them to come and look at his mice. And I suspect that when the husbands arrived from Philadelphia and Boston at the end of the week, all sorts of checks had been written. Anyway, he had a very good way of getting money for his work. And it wasn't until he met Roscoe Jackson, who was an automobile magnate, and gave him enough money to build the actual building that he really took off. At which point, he started to breed mice. And the 57th mouse from Abby's barn was the origin of the famous C57 black 6 mouse. So he's sort of in there from the very get-go. And it's sort of a wonderful story, almost 100 years later. And I'll just go on to mention the fact that George Snell went on to quietly join C.C. Little at Jack's and discover the major histocompatibility locus and ended up with the Nobel Prize. So right now, here's Jack's. It's right next to the Acadia National Park. It's just an unbearably beautiful place to work. We have about 1,200 people at this campus. We have 2 million mice. Don't even think how that goes. We serve about 2,500 labs around the world, organizations, as well as 68 countries. So mice are leaving in large combi trucks every day to go to various places. And we maintain about 15,000 individual mouse strains. So one might ask the question, why? I mean, why would you keep 15,000 mice, not all on the shelf, some of them are in the freezer, but why would you bother with this? And so here comes my pitch for the reason why it's actually important to think about Abby and her collection of mice and why this is now, 100 years later, really become something that we're extremely interested in. And the reason is that we're not all the same. And so when you're thinking about how you're going to model a disease, those of you who are clinicians know perfectly well that there is no one phenotype, as it were, one presentation. There's always some strange outlier, or somebody who is strangely well, even though they have some horrendous predisposition. And so the big question is, if you're going to study disease, how are you going to do it unless you have models that actually mirror the kind of complexity and diversity and variation that we see in humans? We're outbred, obviously, but still, it's important to figure out what is going on. And we know from the increasing information we have from GWAS studies, electronic health records, and the explosion in human genetics, that indeed one of the major problems we have is that science is not simple. Monogenic diseases, even monogenic diseases, have their variability, and that's because disease is not simple, and it's happening in an organism where there are multiple networks that are interacting, and those networks are subject to variation, and that variation produces all sorts of distinct disturbances. And therefore, even if you have one particular gene that's at fault, you're going to do that against a backdrop of all sorts of variability. So as a result, we really have to think in much more realistic terms about how we make models for studying disease in humans. There's another reason, and I mean, this is just, I think, a very telling slide, because it illustrates the fact that the inconvenient truth that pharma companies don't really want to talk about is that most drugs don't work on most people. And indeed, even I've highlighted Alzheimer's just because we've been talking about neurodegeneration for the last few days. 70% of drugs that are actually raised towards these diseases don't work. Quietly, they just don't work. So they work in 30% of the people that are being administered, but what about the other 70%? So we really do need to pay attention to what it is about our patients that makes them either susceptible or resistant to drugs. So here is the situation we are in. We have these marvelous 15,000 strains, all inbred. And they're wonderful because they're homozygous, which means that every mouse is more or less a congenic duplicate of its brother or father or mother. And that means they're all genetically identical. And that means that whatever phenotype they are producing is more or less invariant. And so you can have a control mouse and a mouse that you do something to, feed it something or make it run on a wheel, and you have a perfect control system. And that means that you have a perfect control system. The only problem is that if you're using the AJ mouse, you have a high incidence of lung adenomas as a backdrop. Or if you're using the C3H mouse, you have hepatomas that are a real problem. And of course, then there's the mouse, the C57 Black 6. This guy has a very low pain threshold, alcoholic, neurotic, early sight and hearing loss. They go deaf and blind pretty quickly, and they develop low osteoporosis quite early. So if you have a patient who is alcoholic, neurotic, and then this is the perfect mouse for you to study, but let's face it, this is not necessarily the best mouse for everyone. And so really the question we started asking at the JACS about 10 years ago when I arrived was, how good are these inbred mice as models of this diverse human population? And shouldn't we do something more like this? So this then brings me to the question of, well, what are these laboratory mice and how well are they connected to each other? And do we know anything about their provenance? And we do. Abby worked with 3 fancy mouse strains to begin with. And this was the thing you did back then. You got mice that looked pretty or had strange ears or fluffy or whatever. And in this case, the 3 strains, individual inbred strains that she was working with were the mus musculus, the mus musculus domesticus, or the mus musculus castaneus, and some of the others that were sort of in between different strains that got crossed. Eventually, the European fancy mouse, which had many different phenotypes and manifestations, was sort of condensed into the classical inbred lab mouse strains. It turns out that they're all very closely related to each other. So it's a bit like saying we're taking the genetic diversity of an extremely remote, alpine town where everybody's got the last name Zufre. So anyway, the concept is that we should do something about this because the limited genetic diversity shown here, especially among the classical inbred strains at the bottom, really does, I think, drive home the fact that we're not looking at a particularly broad spectrum of possible genotypes to work with. So at JAX, we have the capacity to do this, and we call them Jedi mice just for fun. And they're wonderful. They're all different. Some of them bite. Others jump out of the cage. Others are unbelievably slothful. So, if you're a mouse fancier, which I am, it's lovely. And I'm going to tell you a little bit about some of the work that we have done at the Jackson Lab with these mice, about 3 or 4 vignettes, so that you get a feeling for what you can do with these mice and how helpful they can be. So just let me explain in a little bit of detail the way in which we've standardized the diversity of these animals. So this was not my work. This is work from several very, very talented geneticists at the JAX, Gary Churchill and Alyssa Chesler, who decided before I ever arrived back in 2015 that something needed to be done. So they chose, as part of a large collaborative group, to take 8 of the strains that were known fairly well and had been used a lot, 5 of which were extraordinarily well characterized, and 3 of which were much more closely related to wild mice. So they hadn't been through Abby Lathrop's barn, the others had. And take those and start breeding them together. So it's a pool of alleles that doesn't change, but the combination is completely mixed up. And when you get to about 10 generations, you then take these animals and you start to inbreed them so that you end up with inbred strains. We call them recombinant inbred strains because they're the recombination of the 8 original strains, and now they're just mixed up. Now, that's not a really greatly diverse population. It's certainly not as diverse as the human population, but we know exactly where each allele came from, and we can go back to each one of these mice and trace every genotype to the phenotype and match them. So that makes it very convenient. Plus, these are all now clonal, so it means you're back to inbred strains, which is great. It means that you can do experiments the way we like to do it, with a control and a test. However, if you're really crazy, you go over to the right-hand side of the slide and you use these diversity outcross mice. These are really wild. So what you do is you take the 8 strains, and now you go crazy and you keep on going, and you never stop. So this has been going on now for, a decade. These mice are completely individual. You can't go back to any one mouse, but what you can do is fabulous mapping, because you have enough population diversity to be able to follow a particular phenotype and find the gene that has produced it. So I'm going to show you a little bit. I'm not going to go into the nuts and bolts of how to do this. I mean, for one thing, most of you probably don't work on mice, but I just want to show you what the power is. And the power is, first of all, a realization right from the get-go that from birth to death, everything about biology is variable. This is what I, was so struck by. And here's an example of a principal component analysis on the left done by Laura Reinhold, one of my brilliant geneticists at the Jackson Laboratory, where you can see that even early on, when you make an embryonic stem cell line out of each of these animals, and then ask what happens when you're following that very early time when you go from a pluripotent stem cell and you start to differentiate into a more defined phenotype, you figure, well, those are invariant processes. This is development. Development is the same in every mouse. Well, guess what? It isn't. And indeed, some of these mice actually have completely different ways of getting to the same ultimate gastrulation. But the timing is different, the genes are different. I was gobsmacked by this. I mean, it goes against my developmental biology roots. It was really a very exciting and interesting moment. And this was all done with transcriptomic analysis. Here's another case. This is work from our own lab. This is the other end of life. This is how long does a mouse live? And now these mice are all living in exactly the same cages with exactly the same food, and yet these are survival curves that are absolutely reproducible for each one of these different Jedi strains. And you can see it's variable. It's really variable. And indeed, for instance, the one in Chartreuse there, the really short-lived mouse, for reasons unknown, that's a mouse that's a combination of two of our collaborative crosses mixed together to sort of, mix things up even more. And neither of the parents have the phenotype that eventually kills this mouse, which is a beautiful and extreme model of vasculitis. It's an extraordinary model. It's the best we've seen. And you could never have predicted it from the way the parents looked. So this is it's a gold mine. It's a candy box of exciting phenotypes. We have one with premature leukemia. We have another one anyway, it goes on and on. And oh, by the way, this is my homage to diversity inclusion. I am showing you how the males and the females are quite different. And indeed, look about that, the Chartreuse mouse for males isn't nearly as bad as the females. And vice versa, there are other differences. So, like I said, nothing is invariant. All right, now I'm going to tell you a couple of stories. I'm going to tell you a couple of stories from my own lab. Sorry, I'm not a neuroscientist. And then I'm going to tell you some stories from the neuroscientist lab that we have at JAX. So this one's about cardiovascular disease. And I actually started this when I was at Monash. So it's kind of nostalgic for me to tell you this story. Very early on, we discovered that indeed there were differences between the way mice responded to a heart attack and the way this is done, as those of you who work on the heart know. You go in and you ligate the descending coronary artery. You can either ligate it permanently, or you can do an ischemia preperfusion sort of model by pulling the string off. After a while, these were permanent ligations. It's quite complicated to do these in these different mice because the coronary artery is also variable, and it comes down the side here. And so I'd had my marvelous technician and postdoc would come into me and they'd say, "You have no idea how weird this is, but we think we're at the coronary artery." But what we managed to do was find out exactly how different these things were. So when you did this on a C57 Black 6, now this is just the straight bog standard Abbey Lathrop mouse, you progress to heart failure. If you did this on a 129 background, the animal was fine until 4 days, and then the heart burst and it was sudden death. Every single time, it was invariant. And when we started to look at some of the cellular differences between these animals, we noted that there were some very interesting changes in the way in which these animals responded at a cellular level. This is beautiful work from Elvira Forte, who was in my lab at the time, and looked at the way in which the fibrotic response to injury progressed over the first 30 days. And so you can see that there's a kind of an inflammatory stage, a proliferative stage, and then finally a maturation stage, which has been well documented by many labs, and this, in this case, just shown up with a nice marker. So what we discovered was that there was a very interesting, very early difference in these animals, and I just want you to sort of have a sense of the variable interstitial occupation of these moments and days after the injury. And what we found was something, because we looked so early, we found a very interesting group of cells which were we called them injury-responsive fibroblasts. We didn't know what else to call them, but they certainly had a very transcriptionally unique footprint. And the reason I'm telling you about those was because we found that indeed between these two strains there was a marked difference in what these cells were producing, and indeed there were many more of them in the 129. And what we surmised after thinking about this and looking at the pattern was these fibroblasts were coming in and making faulty scar. And the scar was all not well put together. And so, of course, when you start pumping blood through something with a faulty scar, it's going to burst. And that's exactly what was happening. So it was the intrinsic response and the elaboration of particular cell types that was obviously encoded in these 129 animals. So Milena Furtado in the lab went on to look at all of the collaborative cross founders and another couple of other ones. And this is just a sort of a snapshot to give you a feeling for how different each of these strains is in terms of the way in which these animals respond. Many of them were okay, that's the blue. The dead ones were the orange, and that was an AFib basically. And then the green is rupture, so that's what the 129s were. And then the black 6 were, in the end, dying from heart failure. So it's not important. The idea is that they're all different. We then decided to do a much more in-depth analysis. And so we decided to go ahead and look at as many possible collaborative cross mice as we could. And so we looked at 32 strains across a 1 month period and took good notes. And what we found were that some of these animals had really unbelievably robust hearts, and they basically walked away from it. Whereas others were just a disaster and immediately started to dilate, and everything in between. So it was a complete goldmine in terms of models of the way in which the heart responds to injury. And here's just some more data to show you that survival was different, ejection fraction was different, LV volume was different. No matter what we measured, it was different between the animals. Well, what that meant was we could go in and start mapping because we had the capacity to do this because we had just enough power to be able to get in there and do something. And we got lucky. And we found that there was one QTL that was quite interesting, and there was one gene called myo18B that caught our attention. This was a bit of looking for the keys under the lamppost, but for those of you who do GWAS, you know you sort of have to do that, otherwise you go bananas. And so we had a look at this myo18B in the literature. It turns out it's expressed in the heart early on. That made sense. It's also very important in the appropriate registration of the sarcomeres in both cardiac and muscle tissue. So knockouts have really disrupted looking muscle. We were also interested in the fact that clinically there were several different reports of mutations in this gene, which were associated with various kinds of congenital myopathy and cardiomyopathy. And we were collaborating with Damien Smedley at Genomics England and found that, indeed, in his collection, the patients with heart disease had many more polymorphisms in this particular gene. And then Kari Stephenson came up with this very interesting description of a particular set of genes that appeared to be very highly associated with defects in the cardiovascular system. Okay, great. Now we have a gene. Now what do you do? I mean, now we all know what the next problem is, and the next problem is networks. What are you going to do with it? Turns out that myo18B doesn't work all on its own. It's not like, dystrophin, which you can sort of say, well, that just sort of messes up the muscle. Look at all the things it works with. So now, of course, we're into proteomic analysis, trying to figure out whether this is actually meaningful or not. And all I can say is it's a journey, but we've gotten quite far because we're down to a gene. All right, that's the end of that vignette. Now, I'll tell you one more. This one came because COVID showed up. Everyone went home, and I was going bananas because I was thinking, what could we do? How could JAKs contribute? I mean, this is a horrible, horrible thing to happen to the human race. And as you know, COVID is a very complicated disease. It has different manifestations in different people. It affects many different organ systems. It's also very different in terms of its severity, and we saw that in absolute, broad brushstrokes as we watched people get this disease and come down with all sorts of different aspects to it. And indeed, we saw that the young were spared when the aged were not. We saw that there were differences between men and women. We saw that there were some people who never got well, et cetera, et cetera. I don't have to go into detail here. And obviously, we were interested in the host response, because obviously that's what was changing it. This was the same virus. It was affecting everybody a different way. So the big question was, is it genetic differences in the host that's making the difference and making more people susceptible versus less? So by definition, we need a mouse model for this. And so obviously, I don't have to mention why you need a mouse model for this, but obviously it's very useful for diagnostics. It's very important for vaccines and antiviral testing, but it's also good for mapping host genetics. The problem was that the original Wuhan strain did not infect mice. And the reason for that is right here in this graph on the right there. The mouse ACE2 receptor has specific mutations in it that do not allow the spike protein of the SARS-CoV-2 virus to interact with it, so you can't get the virus into the cell. This is a problem. So we collaborated with Stan Perlman, who had made a nice mouse for studying SARS when that came out. In this case, it's an overexpressing human ACE2, and the mouse, of course, and it's on a keratin 18 promoter, so it's expressed in the epithelium, which is where it needs to be. And sure enough, the mice got very sick. But the trouble is, they got way too sick and way too fast. So what you're seeing over here is the survival of the mice after having a reasonably low PFU infection intranasally, and they lose weight, which is the first thing you do if you're a mouse and you're really sick. So clearly, we were maybe going to be able to use these for vaccines, but it wasn't a very good model for all the other things we wanted to do. And we knew that the collaborative cross mice were particularly good as models of other diseases, including West Nile, Ebola, etc. And so we thought, well, maybe there's a way we can play this game. And so what we did is we took the original model and we just crossed it into the collaborative cross and looked at the F1s. And so the question was, now that there was a transgene in a different genetic background that afforded infection by this virus, were we in a position to be able to see differences in the way the animals behaved? And they were very, very satisfyingly different. So I'll just show you a few pieces of data here. The one on the left is the susceptible black 6. In this case, that's the original mouse that got sick too fast. Then there was a sex bias where if we went into the NOD founder, we got some animals were fine and others were not. In this case, the females were fine, but we had it the other way around. And then finally, there were some mice, PWK, that walked away from the whole thing, even though they got infected. So, wow, it was exciting, and there was tons and tons of data, and every single mouse had a different trajectory. And here is a sort of overview of all the different ways in which these mice differed from one another. So we had 8 different phenotypes, we had 8 different models. It was terrific. So we immediately put those into the catalog, and off they went. Meanwhile, we had a look with our collaborators out at the Rocky Mountain Labs, the NIAID, who were doing all our infections for us, because you're not allowed to use viruses at JAX. The 2 million mice are too valuable. And what we discovered was there was a massive involvement in interferon, which was not at all surprising, because that is to say, the sensitive ones were really lagging in their interferon response, whereas the resistant ones were just jamming interferon into high gear the minute the virus showed up. And this perfectly matched the emerging picture that the human geneticists were coming up with. In this case, this is Iwasaki's work, and she at Yale. And what she showed was that patients who launched a very rapid response with a high interferon expression were much more likely to survive and to resolve the injury, whereas those patients that had a kind of sluggish response, the trouble is the virus got going, and then the virus started to, get a handle on this host and replicate and cause real trouble. So we were very excited about the fact that we had a model for each one of these different phases of human infection. But by this time, of course, they had a vaccine. So no one was interested. So I said, what am I going to do with this project? And I thought, well, I guess we might as well just look at the variants of concern. And it turned out that the variants of concern could infect mice no problem at all, because they had mutated their spike protein, and now they could see the ACE2 in every mouse cell. So suddenly everything got very easy. We just took the mice off the shelf, we put the virus in, and we looked to see what happened. And it was really fascinating. We found that indeed, here you see, for instance, this is the trajectory, now pretty pretty much of an, I don't think it's working anymore, it doesn't matter. The K18ACE2 was the original mouse, and you can see that Castaneus, which is an absolute bulletproof mouse. I mean, you can feed it high fat and it doesn't get fat. It's sort of our mouse, our super mouse. Well, not when it saw SARS-CoV-2. And we looked at beta, and we looked at gamma, and we looked at delta, and we looked at epsilon. The mice that didn't have any problem at all were very productively infected, but they just shoved it off. And so we had a chance to really dig deep into what was going on with these animals. And I won't go into the details, but they're essentially infecting different cell types. Each one of these viruses infects a different cell type in the mice, and it does so in a very different way depending on the host genetics. So I don't know exactly what it is that's making the Castaneus mouse so susceptible, but what I'd like to suggest is that we're going to have a mouse for every variant of concern for any pandemic that comes up. We just have to be ready for it. And so we're sort of the NIH has a large pandemic preparedness program, which we are part of, and we have now a very active outbreak science program, thanks to SARS-CoV-2 of all things. Finally, obviously the big one right now is long COVID. Is there a mouse that'll tell us what happens in every person who gets long COVID? And as you know, it's all over the map. Some people get very sick, some people just feel tired, and obviously we have mice that we can now use to address these problems. All right, that's a quick vignette about SARS-CoV-2. Okay, let's get on to the really interesting stuff, the nervous system. Now I'm going to venture into foreign territory because all of this work is done by people who work at JAX, who are my beloved faculty, and who were nice enough to send me all sorts of interesting slides. So I will try my very best to sound like I know what I'm talking about. All right, now in this case, this is work from Gareth Howell's lab. Gareth has been working on the interesting variation in the response to Alzheimer's disease and to other neurodegenerative diseases. In this case, I'm going to give you a little vignette which just shows you how powerful these animals are. In this case, you're looking at a mouse that is overexpressing both amyloid and presenilin. It's a known transgenic model that gets Alzheimer's or is predisposed to Alzheimer's. But he asked the question, what would happen if he jumbled up the background? And so he did exactly the same experiment we had done with COVID, and he crossed it into the Jedi mice. And the question was, are these animals going to be different in their response to the overexpression of this aggravating transgene? And the answer was yes. It's really rather spectacular what he found. So here you have one of these Y mazes. So what happens is you put the mouse in the Y maze, it explores both, and it has spent a lot of time being used to the familiar circle. So you give it a novel cross, and you start the mouse, and it explores both. You wait a 30-minute delay, and then if the mouse is behaving like a regular mouse, it'll go straight for the novel one, because that's where he thinks he's going to find some cheese. And so what this story is right here, in terms of an outlier or a group of data, is the C57 black mouse starts and goes to the familiar mouse, to the familiar Y maze, then goes to the novel Y maze, but doesn't really get it, and doesn't, so pretty much of a loser. And Castaneus is also a bit of a loser in this case. But you can see that WSB is a very good mouse at figuring out how fast to go to the novel side of the Y maze, and with the transgene loses that. So it's essentially the prediction of loss of cognitive function. But look at PWK. PWK couldn't give a toss. It's basically fine. It doesn't care about presenilin or AP, because you can just imagine how exciting this is. And of course, he's interested in microglial biology, so we've now gotten into looking at how the microglia are different in the PWK. And of course, the idea of having a model that is so resilient to some of the greatest risk factors in Alzheimer's is extremely exciting. He's also gone on to do single-cell sequencing of the Jedi microglia, and I'm not going to go into all the details. Invite him for a seminar. He's a fantastic speaker, but he has found extraordinary differences in both the abundance and the kind of genes that are being expressed, either in the interferon responsive or in the disease-associated microglia. These are just single-cell analyses showing you which genes are expressed and how different mice are being activating different genes in the different cell types. So this is an ongoing experiment. We're trying to figure out what it is about those PWK mice that can make them so resilient, because obviously if we can figure that out, we might be able to do something about it. Oh, and this, so when I said to Gareth, could you send me something on that beautiful story? And he said, yeah, I will. And then his sidekick, who's a bioinformatician, said, oh, do they work on epilepsy? And I said, yeah, they do. Well, can I send you something? And I said, okay. So this is really preliminary, but he said, tell them it's preliminary. I said, okay, I will. So Matt Mahoney sent me a potential new model of a tuberous sclerosis complex. It's one of the dominant autosomal epileptic conditions. And what he's shown is that if you put a TSC mutation into a normal mouse, you end up with no real phenotype. And that is to say the C57 is a terrible model. However, if you do that same experiment by crossing it into one particular mouse called BXD87, it's another of our Jedi mice, all of a sudden, the mouse, even though the parents didn't get epilepsy with this particular transgenic amplifier, now is suddenly producing a very, very responsive epileptic phenotype. So I don't know if that's interesting to anybody, but if it is, call Matt, or I'll tell you how to get in touch with him. All right, finally, I'm going to tell you how we could potentially make this more accessible to people who don't want to have 600 mice on the shelf at any given time, which is unfortunately what you need to do the big mapping experiments we do. And the story is actually a beautiful story that was headed up by Martin Pera, who used to work here in this precinct, as you recall, and who followed me over to Jack's because he got interested in the idea that we might be able to study genetic variation in some of his most interesting neurodegenerative diseases, one of which was the DYRK1A mutation, which gives severe autism and Down syndrome. And indeed, he very quickly discovered that if the DYRK1A mutation, it's just like a haploinsufficiency. If you have one lacking, you end up with a very early lethality in C57 Black 6. So it's a good model, but it's kind of, again, too far, too fast. In the 129, it seems to be fine. It's a resilient strain. So the question is there a therapeutic target there to grab? And so he's been working at the level of cells because he's the master of stem cells. That's what he does for a living. And so he'd made cells out of, with Laura Reinhold, our stem cell guru, he made stem cells, embryonic stem cells from C57 and from 129. And then he asked the question, if we inhibit DYRK1A either by chemistry or by genetics, what happens? And the answer was that in the case of the C57 Black 6 stem cells, the neural progenitors proceeded to a neuron and then they disintegrated very, very rapidly afterwards, which is what you'd expect from an autism spectrum disorder. The coolest thing was that there's been a model of DYRK1A insufficiency that's been floating around for some time. And in this case, it's a cell line that has a particular name called CULF2. For those of you who are autism people, you'll probably know all about it. And what he discovered was exactly the same thing happened in those particular cells, which were from a DYRK1A patient, a DYRK1A mutant patient. And so what we think is going on here is that we're essentially, it's just not going on. Just give me a second. I'm getting your visitors thingy in here. there we go. What he's done is a ton of transcriptomics and electrophysiology and imaging, and I'm giving you the party line, the summary, which is BDNF is impaired, and that's what's going on in the animals that are susceptible, whereas in the 129s, it appears that BDNF is active and fine, and it literally spares you from the neurodegeneration. And again, this is a summary of the work that he's done. He's published this. It's a beautiful study in which he shows that it's actually the C57 black 6 mouse cells are switching the neurons to make a inactive BDNF receptor, and that's as opposed to the 129, which is actually seeming to maintain an active BDNF receptor. So the hypothesis is that if you could reactivate BDNF, somehow you might rescue this phenotype, and of course that's what we're now thinking of doing, and of course if it's in cells, you can screen for drugs a lot easier and more cheaply and quicker than you can in mice. And just to give you a feeling for how exciting this was to Laura, she's prolific, she went back and said, well, let me just make a whole bunch of cells. So now we have about 600 different lines with cells that are associated with them in the embryonic and or induced pluripotent state. So if there's something that you want to do on this kind of a platform, it's now going to be something that we can hand out. And it's very exciting to imagine that we're going to be able to really take the genetic diversity out of the animal and go into the dish and use it for mapping or drug discovery. So here's just our sort of vision of how this is all going to work. You can imagine engineering candidate mutations into these cells. You can differentiate them because that's what we can do with stem cells. You can match mouse and human cells because we're also making a whole bunch of human cells. And eventually you can hopefully use this to go much more deeply and quickly with the defined genetics in the mouse system to potentially find targets. And so this is why we think that the cell platforms are going to be really super exciting for the future. All right, so I'm sort of at the end of my tale. I just want to make a few comments. The first comment is, I really believe that there's something sort of profound here. If we are going to be the purveyors of models for human disease, we really do need to make the models as diverse as the people that we are trying to study. And obviously, that's something that we're excited about, and we kind of imagine it working something like this. Imagine a cube full of data about humans. Now imagine that in the mouse, you can get much more data, much more dense data. Now, it's not all going to be relevant, obviously, because we're not mice, but there will be aspects of this that we can extract out of that data set and essentially superimpose onto the human data set and fill in the gaps. So that's a sort of a way of pictorially imagining the fantasy we have about this. And if that's the case, then you can take all of this work that's being done right now in the human genetics setting, where human samples and biobanks and electronic health records are telling us tons and tons about what the associations are between genotype and disease, and do a separate sort of investigation using a much deeper and more extensive way of getting to phenotype, because you can cut mice up. And as a result, you can then hopefully use that data to really refine what it is that we're going to focus on in the human. It's a fantasy, but you know, you got to have dreams. So in this case, I'm envisioning something where disease will be predictive, but then you can actually validate it. You can validate it in cells, you can validate it in mice, you can go in and say, if that's the smoking gun gene, let's wipe it out, or if that's the regulatory region that's producing a hypomorph, let's play around with it, and actually start getting face validity for some of these projects that we're so excited about. Anyway, that's it. I'd like to just very briefly spend 2 seconds saying that my lab team, and I won't go into all of the people and the collaborators, are obviously behind everything I told you from my lab, and I mentioned the faculty, but I have many, many more stories to tell. And finally, the mice. I mean, you can't do this without an incredible infrastructure, and I'd say that JAX has been an enormous enabler for our capacity not only to develop these extraordinary panels of mice, but then to take that to the in vitro and in silico levels. So thank you very much. That's what it looks like. There's our lab down there, and this is about 2 weeks ago, gorgeous colors, and full moon. Please visit. It's beautiful. Thank you.
Thank you very much, Nadia. That was just fantastic. And Nadia's kindly agreed to take some questions. So while you're thinking of some questions, just a couple of observations, if I may. I think you've shown us really that there's extraordinary strength in diversity, and I think that's probably a message that many of our politicians could probably take on board at this time in the world. But you've also shown us the power and the joy of curiosity in science, and the story resonates very closely with me because I spent probably 2.5 years of my life pursuing a serendipitous discovery early in my PhD, which was absolutely frustrating at the outset, around inbred rats, and the finding that I was tasked as a new PhD student with trying to generate retinopathy of prematurity in an inbred rat strain, and this damn strain would not get retinopathy. And my PhD supervisor was very, very diligent researcher, and she said, "You must be doing something wrong." So I repeated the experiment, and no retinopathy. And I managed to convince her to allow me to get an ethics approval to look at many different inbred strains, and that led to a 10-year program of research looking very much into microRNA determinants of susceptibility to retinopathy. So
you
really,
drank the Kool-Aid
I drank the Kool-Aid, and I guess I'm here as a consequence. So the story resonates very closely with me. We have a first question, so I'll leave it to you.
Nadia, fantastic tour de force of a presentation. I would like to kind of come back to your vignette on heart failure, a great model of heart failure with reduced ejection fraction. But one of the things the field has in progress is developing a clinically relevant model of heart failure with preserved ejection fraction,
right,
which contributes to the other 50% that has really bad prognosis clinically. I could see your infrastructure playing a huge role in developing a clinically relevant model with age cohorts with multiple risk factors. Have you made any strides in developing a mouse model of heart failure with
yes,
preserved?
HFpEF. This is heart failure with preserved ejection fraction, and it is an increasing scourge of older women who are obese, among others, but it has more and more relevance as it's associated with type 2 diabetes and other comorbidities. And it's I think most cardiologists are really very concerned about this. And of course, the first thing we did was to look across all of our mice and see if we had a HFpEF model. And so far we do not. Either the mice are fine or they get dilated or their hearts blow up. So there are a couple of protocols that have been published that supposedly induce HFpEF in animals, one of which was Joe Hill's famous high-fat diet plus L-name protocol. I can tell you that that's a bitch of a protocol. We have tried to reproduce it, and several other people, and Joe knows this, I'm not saying anything behind his back, and we think that there may be some really undiscovered problems with the protocol in the sense that it has not been well defined. So it turns out that, you have to sing Showboat and do it on the phase of the moon and et cetera, but really the main issue is that L-name is extremely, extremely unstable. So people don't know this. And so we've gone to great lengths to try to see if we can get this particular protocol to work, because we really would like to be able to run all these same experiments across genetically diverse mice and find out what is it that's really controlling HFpEF and how can we avoid it. All I can say is tune in about 2 months. We should have the answer whether we can actually make it work or not. And I think one of the major problems, of course, is that mice are not particularly good at being sedentary. They don't actually overeat that much. They love to exercise. And so everything about HFpEF isn't really a mouse's normal, the kind of comorbidities are not particularly characteristic of mice. So we may need to go for some real sluggers. We have one called NZO that literally gets fat by looking at a cookie, and we're hoping that maybe that mouse will be the one that will really hopefully give us the protocol we want. But thanks for the questions. Great. It's a real problem in the field.
Hi, a couple of questions. The first relates to phenotyping. A lot of the phenotypings that you're looking for can be quite overt, but some of the things that we want to be looking for example, autism, etc., are going to be quite hard to see. So the first part of the question, I guess, is what's the sort of phenotyping looking for things? And then the second, I guess, everybody's talking about is AI. I mean, the richness of the data that you're getting is extraordinary. Is that starting to be integrated into the workflow?
So I was thinking of putting in a really cool video, and I didn't want to just drive you guys crazy with more data. We have a program which I'm very excited about, which is based on the idea that to really get deep phenotyping of mice, we have to not just do, grind and bind and look for, differences and complexes. We need to actually use the fact that the mouse is alive and is manifesting extraordinarily complex behavior. So Vivek Kumar is another wonderful faculty member, who is also a bioinformatician, has developed algorithms that allow us to have mice in a home cage with very, very little intervention. So we've got all sorts of ways of feeding and watering them without taking them away from the home cage so that their stress levels go way, way down. And these home cages are about to be marketed sometime in November by Jax. But in the meantime, the mice sit in there, and we have cameras that are taking continuous video, and we all do this. I mean, this is the open field test, et cetera, et cetera. The difference here is that Vivek has arranged it with his people that he has coordinates, about 7 coordinates on the mouse. So head, shoulders, paws, something on the tail, et cetera, that the video will reveal the algorithm can actually take those data, and find the most unbelievably subtle phenotypes. Because you can look for rearing, you can look for grooming, you can look for social interaction, because you can do this. It's a very sophisticated program which allows you to not worry about occlusion with one mouse going over another, which is always a problem when you have more than one mouse in the cage and you have the cameras. And the kinds of data we're now getting are really exciting. So I'll just tell you one tiny little story. So John Backman, who's a postdoc in my lab, thought what would be really interesting would be to figure out what it is about a particular project he was working on that would predict when a mouse dies. So we've been looking at the nasty effects of immune checkpoint inhibition on myositis and myocarditis in cancer patients, which is a small but rather lethal comorbidity that the anti-PD-1 gives these patients. And what you really want to be able to do is predict the patients that are going to respond by getting very sick and often dying ahead of time. So you don't give them anti-PD-1, you come up with something else. So we're looking for biomarkers that will tell us whether the animals are going to get sick on this particular drug or not. And so we've done the same kinds of protocols that patients experience when they're undergoing these kinds of immune checkpoint inhibition therapy. And what we find is that right after the fourth administration in a particularly susceptible strain, so it's highly artificial, I have to say. It's very, it's a very complicated genetic strain. The mice suddenly die, and we can't figure out what they're dying of. We looked at everything, and the hearts look okay, everything looks okay. So it's very hard to say, well, that mouse is going to die right now, and obviously we want to be able to predict whether that mouse is going to die well ahead of time. So we have to get some kind of biomarkers. And this extraordinarily sensitive algorithm, which is AI-based, is giving us that kind of information. And indeed, we just last week came up with a particular set of behaviors that are consistent with the animal getting sick and dying. So it's very exciting. And so, I mean, that's the sort of thing that Jax spends a lot of time thinking about. We have an entire mouse phenotyping service, which essentially goes through everything from clinical chemistry to behavioral tests to MRI, to try to refine the capacity to pick up autism in a mouse. And I'd be delighted to talk to you more about that, because obviously Martin Parr is very excited about this project. Sorry, that was a bit of a long one.
Next question. I can just envisage that Alexa's going to be warning us that we might be about to die, and
I can.
playing the death march.
Given how long Nadia took to answer that question, I think I'll defer it to dinner. Because it's
I think that's a compliment. I think that was a veiled compliment to say how extraordinary your command of such a broad array of topics is, Nadia. Tony, did you have a question?
Well, if there's time, thank you for a wonderful lecture, Nadia. I was wondering, in terms of diversity, you mentioned genetic diversity. So the other side is environmental diversity. And do you play around with that, in terms of gene-environment interactions, things like diet and driving microbiota differentially under these different genetic backgrounds, and what happens?
Yes. No, I'm kidding. What happens is that you can just as easily start predicting the way genes and environment interact with each other using these animals as just doing the genetics with everything else being constant. And indeed, we've changed all sorts of things. You can change home cage environments, you can change diet, you can change social interaction, you can change whether they get exercise or not. I mean, you can imagine, you can change every possible parameter. And as long as you are doing it in a rigorous way, so that you don't introduce all sorts of noise, you can use these mice to map all sorts of interesting interactions, including metabolic interactions and the other one you were mentioning, which I can't remember right now, whether the mouse likes Beethoven. That's for Greg. Anyway, you know what I think? I think that I can smell through my AI receptors the need for a drink in every one of you. So I suspect we should just go outside and indulge.
Nadia, before you sit down and before we thank you all again, I'd like to invite Martin Adams, our board chair, to say a few words of thanks.
Can I just...
Well, what a treat. Nadia's energy and enthusiasm, her sheer intellect and curiosity, she really is a tour de force, and she's a global leader of science in the true sense. And as she's roamed the world in her wake, she's left people enthused to pursue a career in science, and along the way leaves various labs in Rome and the UK, and here tonight, in fact, there's a dinner at the Australian Regenerative Medicine Institute, which you can't attend because you're here. This is your own creation. But it seems no matter where you go, you leave in your wake an institute, and more importantly, you're enthusing a whole new generation of scientists. And I just know, talking to the people here the last couple of days as part of the panel you've had, they've probably started with a bit of trepidation, but how's this going to be? And they've walked away with an infusion of energy and enthusiasm. And yesterday you gave a wonderful lecture at Newman College called Forever Young. And the ultimate curious person, you shared your DNA structure with us and the various genes. But I think there's much more work to be done to figure this out, but I think you're a living example of Forever Young. And it's just wonderful to have you here. So thank you to Allan and Maria Myers for making this all possible. Nadia's not by herself. She's a team of 8 of our panel members. And they're all extraordinary in their right. And I just wanted to say special thanks to you, Sam Berkovic, for taking on Trevor's call. And no one can say no to Trevor. But when he asked you to task you with leading the scientific review, it's been a terribly important process. And we're looking forward to getting the findings next day or so. Brendan Murphy, thank you for joining us. You're a very busy man. And to spend 3 days with us is a real treat. Greg Lemke, coming all the way from La Jolla. And I've been there. It's a beautiful place. No one would want to leave there. But great to have you here. Jayshree Kulkarni, who's not here with us tonight, but she's been a huge contributor. Peter Schofield, thank you. Andu, coming all the way from across the road from Weehai. Wonderful to have you with us. And, of course, Allan Finkel, but mostly you, Nadia. Thank you so much. for gracing us yet again. And just because we want you back again, we have a small gift for you. Where? Oh, here. Sorry, I got too many things. This is why I'm the last act. Make everybody else look good. Right. What's the order of play here? Nadia. Would you like to?
Oh my God!
This is to. When you go and see your mice, they say you've had a good trip. And then here, just a small little something about it to remind you, and also for Allan to remind you, in us, that you were here with us.
Oh, how wonderful!
So thank you so much, and
Thank
everyone
you.
else. I think this ultimate advice was, what was it, your receptors that said a glass of wine?
Yeah.
Okay. Thank you all very much, and thank you again. Wonderful.